Hello guys. I guess from our previous video we studied. We saw about part of asexual reproduction. We were majorly focusing on this fission and binary fission and the types of binary fissions. So now let's see the remaining parts of asexual reproduction. This tutorial consists multiple fissions and fragmentation, regeneration, and all these things we will be learning from this tutorial. Now let's see what is multiple fission. I told you what is fission in general, right? Fission means from a body cell you get either two or more daughter individuals. So that is what in general fission means. A multiple fission means from the parent body you get many similar daughter cells. So that is what multiple fission. Now, what happens in multiple fission? From a single parent, you get many daughter individuals. Let's see what process actually takes place inside this mother cell. So this is the single mother cell, right? So this is a single mother cell and has only one nucleus. So first, this nucleus will undergo many nuclear divisions and continuously produce lot of nucleus inside the cell itself. And once it has produced, and during this nucleus division, the cytoplasm inside it, right? You know what is cytoplasm, right? Cytoplasm is a place where you have different organelles, and all these organelles do a lot of different functions, right? Remember? So the cytoplasm does not undergo any cytoplasmic division until this mother cell plans to divide as many nucleus as it requires. Once it has divided, once all nucleus has been replicated by the mother cell, then the cytoplasm will equally divide into as many part of as that of nuclei, which means like you have around 10 or for example you have 8 or 2 4 6 8 and 9 nucleus have been divided inside the mother cells, and this 9 nucleus again will encircles with cytoplasm and equally divide into 8 or 9 new daughter individuals, which looks like its mother. Now you understand what is this multiple fission, right? So this kind of fission, which means inside one mother cells you get lot of nucleus will be continuously dividing and replicating. So this kind of process is known as repeated fission, because through this process you get equal number of new daughter individuals, right? Like as many as nucleus it has replicated, so you get as many as new daughter individuals. So this process is actually generally known as repeated fission. So the best example is the protista, which is verticella. I've also given a microscopic picture of verticella which undergoes this repeated fission. Look at this picture. So if you look at this picture, all of them are verticella. So if you look at inside the picture, you can see a lot of nucleus have undergone many nuclear divisions and have formed many nucleus inside it. Until they are ready, this cell will not undergo the cytoplasmic division. Once everything is completely done, then each nucleus will develop cytoplasm and will form a new individual. This is how it actually takes place. Now we have seen what is multiple fission. Let's see further more about this multiple fission from different organisms. Let's see how multiple fission takes place in Plasmodium. So it's very interesting. Let me tell you what is Plasmodium. Plasmodium is a protozoan that causes diseases in humans. If you can recall, malaria is actually caused by this parasite. And let me tell you how this parasite multiplies inside our body and in mosquito. So this Plasmodium undergoes multiple fission two ways. One is through asexual reproduction, that is from humans, and one is from female. One way it undergoes sexual reproduction through mosquitoes. What happens? These sporozoites, right? Let me tell you what is sporogony. The process in the mosquitoes, inside the mosquitoes, the Plasmodium does this sexual reproduction, and through in its oocyte stages, and produces sporozoites. Sporozoites means daughter individuals. This process is known as sporogony, and all this process takes place inside the mosquito. Through sporogony, once sporozoites, I mean many daughter individuals, has been released inside the mosquito, which means the Anopheles mosquito, which is responsible for this malaria. This sporozoite will be introduced into humans through bites, mosquito bites. That sporozoite then becomes schizont. This schizont again undergoes multiple fission through asexual reproduction, because here you get both germ cells, gametes inside the mosquito, but here it undergoes asexual reproduction once this sporocytes through mosquito bite is being introduced into human blood circulatory system. And then it becomes schizont. This schizont again undergoes this multiple fission and produce merocytes, and this process is known as schizogony. Now you understand how this plasmodium does this, reproduction in different way from different host. You know what is host, right? Host means any organism that provide supplements or place for any parasite to grow. So this plasmodium is a parasite. So in this case you have two hosts, one is mosquito and another one is human. So as you look at this image, you have they have given an asexual reproduction that generally takes place in humans. This plasmodium, once this porozoite has been introduced into the human, it becomes schizont. This is what schizont cell. So then it just continuously produce many nucleus. We collectively call them nuclei. Then all this nucleus will again grow into new individuals, and this process is what we call schizogony. And once this has been formed, the cell wall, the schizont wall will be ruptured and produce this daughter individuals, and these daughter individuals are known as merozoites. Now you know what is how this plasmodium does its reproduction in different way. Now let's see amoeba. This is quite interesting. This amoeba actually does lot of asexual reproduction in different way. Previously we saw simple or irregular binary fission, where one single amoeba becomes two individual amoeba. In any direction can be divided into two daughter individuals. Now in here, this amoeba does this multiple fission in a different way. When there is an unfavorable condition, for example, if that amoeba species is facing any threat due to environmental condition, for example, if there is an increase in the temperature or decrease in the temperature, or scarcity of food, which means there is no food or lack of food, it leads into this process, this phenomenon known as encystment. Let's see how this phenomenon takes place. Let me tell you what is unfavorable condition. If you take any organism, it requires a favorable condition to survive, to reproduce, to grow, to mature. So there are organisms like amoeba and many organisms, when they face this unfavorable condition, if there is no optimum environmental conditions, they undergo a different process to make sure that it lives in the next generation. So that's what's going to happen here. So during this unfavorable condition, which means no food or increase in temperature or decrease in temperature, it withdraws its pseudopodia. Do you know what is pseudopodia, right? Pseudopodia is the major thing that is responsible for migration and eating food, all this thing, catching food in the amoeba. So it withdraws, which means it just takes away its pseudopodia. Let me try it here. So this is how amoeba does not have a fixed shape, right? So this is how it looks like. This is known as pseudopodia, right? I don't want to write it here because it's going to take a long time. So wait, this is known as pseudopodia. It just withdraws its pseudopodia, which means like it just takes away its pseudopodia and secrete a layer. For example, it already withdraw its pseudopodia, and it's going to secrete a layer now. Let me draw it. So it just withdraw it. It's going to secrete a three protective layer which is made up of chitin. which is known as chitinous cyst wall. So it just takes away its pseudopodia and develops a three protective layer around it. And this three protective layer is actually made up of chitin. So and it becomes inactive because the condition because the condition is not favorable, right? It is unfavorable, so it just becomes inactive. And this process what we call is encystment. Now you understand what is encystment, right? Now let's see more about how why this encystment phenomenon takes place. Once this cyst has been formed, wall around it has been formed, inside the cyst the nucleus will continuously divide into many nucleus. These many nucleus we call nuclei, dotted nuclei. So what you have a protective layer because the outside condition is not favorable, but inside it the replication of nucleus takes place and you get dotted nuclei, which means many nucleus inside it. And when the condition becomes favorable, what happens, this layer around it, right, the chitinous cyst wall will be broken down. And you know what happens, it absorbs the water and breaks the layer around it. And inside it, I told you all the nucleus have replicated and you get a lot of daughter nucleus, right? This will grow into new individual inside itself. Those new individuals are called pseudodiaspores. Sorry, pseudopodiaspores. Pseudo, sorry, pseudopodiaspores. Diaspores. Pseudopodospores. Sorry, there is a problem with pronouncing that word. So this pseudopodiospores, when the condition becomes favorable, the cyst around the amoeba will absorb the water and breaks down, and this layer also will breaks down, and all this new daughter individuals are called amoebae, young amoebae, will move out of from this amoeba and grow as a new individual life, lead a new individual life. And also it develops its pseudopodia, which is essential for its migration, right? So this is how it actually does multiple fission during unfavorable condition of this amoeba does this asexual reproduction. Okay, now we have seen what is multiple fission in amoeba during unfavorable condition. Let's see more about uh, again multiple fission in other different organisms. So strobilation. Some metazoans, right? Some metazoans are it's a kind of different. It's an animals that have a very simple organization of tissues have organized into the different to do different perform and different process. So these metazoans animals do this strobilation process. Let's see what is strobilation in detail. So it actually does this transverse fission. By through that you get a lot of new daughter individuals. They have given example Aurelia species, which is also commonly known as moon jellyfish, which lives in the ocean or sea. So this moon jellyfish actually does two kinds of reproduction, which is asexual and sexual reproduction. But what we are going to see here is asexual reproduction. But in order to understand this strobilation, it is very important that you understand asexual reproduction too. So that is why I have given an example picture here. But let's see how this transverse fission takes place. So this is that polyp or a stabile. At particular point it just ceases to grow its tentacles further. Instead, it just continuously produce many daughter individual through this transverse fission. All these are many daughter new individuals. Let's see how this process takes place in this jellyfish. I mean, sorry, in this yeah, it's a jellyfish. environment. Let's see its life cycle, then you will have a clear picture and clear idea about it. So, this is a matured moon jellyfish or Aralia. So through sexual reproduction, it produces a larva, and this larva grows and becomes a polyps. Once this larva, what happens? This larva moves around and grows, and it looks for any hard substratum to attach. Once it has attached on any hard substrate means any floor like sea floor, or you have a lot of different organisms that provide a substratum for many organisms. For example, corals. Those likewise some rocks, all this also called substratum. So once this larva finds a substrate to attach, it just grow as a polyps. Okay, this structure is known as polyps. So it grows slowly and develops tentacles because it has to feed, right? This tentacles helps in the feeding process. It just filters and gets all the zooplanktons, I mean all the microscopic organisms that it gets, it just eats it, and it develops. At certain points, it ceases to develop its tentacles and undergoes this transverse binary fission. You can see the difference between these two polyp, right? And it just suspended in the water. It was suspending in the water, and once it has found a hard substrate to attach, and it grew as a polyp, and this polyp eats and grows to some extent, and then it ceases to develop its tentacles because it wanted to do the transverse fission. So here you can see many new daughter individuals have been formed slowly. Once this has been formed, as many daughter new individual as it wants to do, and it undergoes a process known as strabila, where this many daughter new individual produced through this transverse fission will get separated from the mother, I mean from this polyp. It just moves around it. You can see like one this is known as, I mean, the individual that is separated through transverse fission in Strobilation is known as Ephyra. So this Ephyra, you can see many Ephyra is like flying away, I mean separating from the polyps, I mean the grown strabila. So now you understand how this Strobilation takes place in metazoan animals, especially from some jellyfish, molefish. Okay, now we have seen what is Strobilation. Let's see more about multiple fission in few other organisms. So in our previous slide, we saw what is Strobilation, right? Now let's see what is Plasmotomy and sporulation. Let's see what is Plasmotomy. What happens in Plasmotomy? These many giant amoebas will contain many nucleus inside it. We call many nucleus as nuclei, right? So this multinucleated parent again produce a multinucleated daughter, and this multinucleated daughter then does this nuclear division to maintain proper nuclei. So let me give you with a picture. This opalina is a protist that actually has many nucleus inside it, right? If you look at it, many nucleus, so we collectively call them nuclei. And when it reproduce, the daughter cell also will have many nucleus. So it will again, the many nucleus will do nuclear division to maintain proper nuclear count. So you can also see that Pilomaxa also does the same plasmotomy multiple fission. So this is the microscopic picture of giant amoeba Pilomaxa. You can see different, I mean many nucleus inside it, right? So when it reproduces, the daughter cell also will have many many nucleus. That's what we call multinucleated daughter. So now let's see what is sporulation. Previously we saw that amoeba does this encystment when there is an unfavorable condition. So similar, but that's a different process. But here, this during sporulation, amoeba again faces unfavorable condition, but it does not do this same process, encystment. So I told you in unfavorable condition it also does encystment. Here it is undergoing another different process which is known as sporulation. Let's see about that in detail. Sorry. So what happens, the amoeba cell during this unfavorable condition, the nucleus inside the cell will breaks down into small fragments or chromatin blocks. You know what is chromatin, right? Before reproduction, any cell will be in its chromatin forms, a thread-like structure, right? Remember, you must have studied in your lower classes. So this is how it looks like. See, there was single nucleus first, and the nucleus has broken down into many small fragments, and all these fragments again will develop a nuclear membrane, and also will be surrounded by a cytoplasm and develops a spore case. This is a spore case if you look at it. So look at here. You can see the spores here now. All have developed a spore case, which is filled with cytoplasm and the chromatin blocks, which was first broken down. And when the condition becomes favorable, the parent body disintegrates, and the spores will liberate from the parent body. You can see it here. So all these individual spore case, right, all of them have fragments of nucleus and have developed a cytoplasm and a spore around it. And when the condition becomes favorable, the parent body disintegrates and all of them will come out. Again, the spore case also disintegrate, and the new amoeba hatches and develops a lethal individual independent life. So these small ones, we call it as hatchlings. All these things that comes out of its parent body right after the disintegration are called hatchlings, and the spore case also just disintegrates and new amoebatic cells will come out of it. Excuse me. So now you know what is sporulation. Now let's see what is budding. So far we were seeing about fission. From fission you have seen multiple binary fission, and in binary fission, based on the plane of division, we have different binary fission. Then we also saw multiple fission. It takes how it takes place in different organism with in different process. So we were very thorough with that fission. Now let's see what is budding. So from the parent body, you get one bud or more buds, that each buds will develop into an young one. Okay, so parent body. So on the parent body surface, you get a bud. The buds grows. Buds are in simple you can say it is a projection or an outgrowth that starts growing on the surface of the parent body. It can produce one bud or more buds that will have the potential to develop into an young one. So let's see with the example of sponge. So you can see here, this is the sponge. It has an outgrowth, this bud forming, and it grows slowly. Once it has reached its maximum level, I mean the optimum condition, the bud constrict in the middle and detach from the mother and grow as a new individual. So this simple process is known as budding. Again, this budding, based on the place where it actually takes place, whether it is inside or outside the body, it is again classified into endogenous and exogenous. Let's see what is exogenous budding. Exogenous budding, this was actually, I mean, this exogenous budding, they have given an example with hydra. Let's see how it actually takes place. So when the this exogenous budding takes place outside the body surface. That's what I told you, right? When the when the food is available plenty, so this hydra, hydra develops outgrowth or projection from the ectoderm. Ectoderm means outer layer, and the ectoderm starts growing on the ectoderm. The cell starts growing and starts forming this bud. And both ectoderm cells, I mean from cells that is present in the outer surface of the body, and the endoderm, which means endoderm cells, from the inside the body. Both ectoderm and endoderm cells push outside to form this bud, and then this bud is actually connected through gastrovascular cavity through lumen. Lumen is a tubular structure because this bud has to get all the supplements to grow, right? So through that gastrovascular cavity, which is connected to its parent body, it gets all the supplements and keeps on growing and forms these tentacles. Once these tentacles and it has grown grown into an optimum condition, it just does it constrict in the middle here and detach from the parent and grow as a new individual. Now you know what is exogenous budding, right? Now let's see what is endogenous budding. Endo means inside. So this takes place in Noctulica, which is a dinoflagellate, marine dinoflagellate again. So what happens inside this Noctulica? Hundreds of buds forms inside these Noctulica, but not all the buds that is formed due to this budding gets out of the cell. Only a few individual gets out of this cell and grow as a new individual, new daughter individual, and repeat the life cycle. I have given an example picture. Now, this is how it exactly look like if you look at the microscope, a single Noctulica species. And this is also a dangerous species, but let's not get into that topic. Now we have seen what is budding. Let's see there are another process which is known as gemmule formation, which takes place in both sponges from marine water as well as from fresh water. What are these gemmules? So let me give you an example. See, you have the sponges here, right? So inside the sponges, let me draw an image. Okay, this is sponges. Okay, the substratum. So inside these sponges, there will be a lot of internal buds will be growing, and these internal buds are known as gemmules. So these gemmules are very hot. When the condition is not favorable for the sponges, the parent body slowly disintegrates. And let me draw it. And now you have all these gemmules, and these gemmules will and this gemmule continuously grown gemmule will have a food laden archaeocytes. Inside this gemmules you have food laden archaeocytes. Archaeocytes are nothing but the amoeboid kind of cells which have different function inside the cell, inside these gemmules. So this grown gemmule, it can withstand any adverse condition. Adverse means worst environmental condition, and it just floats around the water. And when the condition becomes favorable, it just goes, it just go and attach and grow as a new individual. So now we have seen what is gemmule formation. Let's see more about other process in asexual reproduction. We have fragmentation. Let's see what is fragmentation. So as you from the name itself, it's very easy for you to understand, right? Fragments. The parent body breaks into fragments. The parent body, for example, this is the parent body. I can give you a simple example from cyanobacteria, which is blue-green algae. So this is how exactly it looks like. So this is the parent body. The structure of the I have not clearly drew it properly, but this is how it actually looks like. See, if you look at this structure, this is the entire body of that blue-green algae. If it is about to reproduce, what happens? It just breaks into fragments. So this, right? Let me try it. This is a fragment. So, and each of this fragment that has been broken down from the parent body has the capacity to develop into a new individual. For example, it will start growing new parts of the body. See? Again, this will also grow like that. It will grow. So you have fully grown individual from a piece of the parent body. Now you understand what is fragmentation? Okay, fine, clear. Now let's see more about fragmentation in different individual organisms. So this pedal lacration happens in sea animals. So do you know what is sea animals? I believe that you guys, all of you must have seen this Finding Nemo, where this clownfish goes and hides inside that, tentacle-like structure, which just keeps on moving here and there, right? Those beautiful creatures are called sea animals. This species can able to move slowly. What happens? This paddle accretion means, I have given an example. Let me see here. So this is that paddle. So what happened? Paddle accretion means the paddle lobes, right? This is called lobes. While moving, this sea anemone, it just breaks or leaves its part of its foot, which is the paddle, and this paddles again can grow into new individual organisms, which is mesenteries, and also able to form tentacles. So from a fragment, from a piece, you get an individual whole new organism which has a capacity to develop tentacles and as a whole organism. This is what pedal lacration in sea anemone, which is one type of fragmentation in sea anemones. Now let's see tapeworm. So you know about tapeworm, right? That is a process known as apolysis. So what is apolysis? Let's see. In tapeworms, this teniosolium, it just keeps on growing, which is present in our intestine, I believe. So this proglottids means it is a uh, part of the I mean very grown part, completely grown part of the uh, Teniosolium tapeworm. So this will be continuously detached from its parent body, these pieces, right? And these pieces, once it has been detached from its parent, once it has reached outside the host, this or parasite, right? For example, if from humans or pigs, it just this grown part of this proglottids, the gravid or ripe part, once it has been detached from the parent, these pieces will has the capacity to grow as a new individual, and these pieces will helps to transfer this embryo to from a primary host, which is man, to secondary host. I believe that you guys know how this parasite transfer from man to pig and pig to man through excretion. Excretion, when this excretion is eaten by this pig, it just gets inside the pig and grow as a new individual. So that is what fragmentation in tapeworm. Now we have seen what is fragmentation. Let's see what is regeneration, the last process in asexual reproduction. So regeneration means regrowth in an injured region, which means a region which got affected. So this was first discovered by Abraham Tremblay in 1740 from Hydra. So he observed this regeneration process from Hydra in 1740. So what happens? This regeneration again is of two types. One is morphollaxis and epimorphosis. Let's see what is morphollaxis. This morphollaxis, where the whole organism grow as a new individual. Let me give you an example with the picture. Look at here. This is the initial stage of the regeneration. For example, this is the whole individual organism. It's just broken into different pieces, three pieces, and these pieces, this part of the hydra, it just has the capacity, which means like it got injured by for example, due to accident, it just broken down into three pieces. The difference between fragmentation and regeneration means this happens in regrowth in injured region, whereas in fragmentation it is like a voluntary process where a piece of parent body will be broken down into pieces and produce an young one. But here, by accident, if a part of an individual organism, for example this hydra, by accidentally it loses its pods. For example, broke down into three pieces, and this pod of the hydra has again will regenerate and grow as an individual organism. Again, if you look at here, this pod also will grow and regrow its tentacle and form a new individual organism. This is the old pod from here and the newly regenerated regrown pod. And this center pod, which got accidentally got separated from its entire from an individual organism, again has the capacity to grow or regrow the as an individual organism. From whole part, you can see the entire cell is growing into new pod, right? And it can also undergo longitudinal through longitudinal incision also it can divide too. Now we know what is morphallaxis. Morphallaxis means if by chance if an individual, a part of an individual organism got injured and if it lose, it can again grow into a complete new individual organism, which we have seen through this example. Now let's see what is epimorphosis. This epimorphosis again is of two types. What is epimorphosis means? Replacement of lost body part. Replacement of lost body part. For example, if it loses fingers or nails, again it will be replaced, not regrown, it will be replaced. Okay, let's see how again this epimorphosis is of two types. One is reparative and restorative. Let's see what is reparative. Reparative means only a certain type of tissue can be repaired. It cannot be regrown back or restored. It can be repaired, like the certain tissues, if it is damaged, that tissue can be repaired, like there will be some modification takes place and that tissue becomes normal again. But in restorative, the severed body part can be replaced. For example, you must have seen in your at your home that wall lizard, most of the time it loses its tail, again after some time it starts developing its tail. So that is what a severed body part can be restored again. And starfish also can, if it loses any of its part, again it can able to regrow. So here you can see it, right? So this is what we call as regeneration. Now we know what is regeneration. Regeneration in simple, regrowth in an injured region. Again, it is of different types. We have seen that in detail. Now, okay, fine. So thank you. As of now, this is about your asexual reproduction. I will see you next in your next week. I will see you guys in next video with sexual reproduction in different organisms. Thank you.